Welcome to Study Loop.
Here for this question, we are looking at the molecule and protein of polyadenylate polymerase. It's a mouthful, but if you break the word apart, you can figure out its function. So poly means many, adenylate means the addition of your adenosine molecules, and polymerase is going to be an enzyme that's going to create a polymer. So this protein, polyadenylate polymerase, is responsible for adding that poly A tail onto the mRNA molecule. So here, if our polyadenylate polymerase is dysfunctional, we're going to cause the removal or the improper placement of this poly A tail. So in reality, we need to consider the function of this poly A tail for the mRNA molecule. So inside of the cytoplasm, where this mRNA will be transcribed into proteins, you're also going to have other enzymes you could call RNases, or say microRNAs. And these are proteins responsible for degrading the mRNA molecule. And this will prevent the overproduction of proteins from the mRNA. However, these molecules would typically start at the end of the poly A tail and would degrade their way up until eventually they reach the mRNA molecule. However, if you remove that poly A tail, they're going to start at the base of the mRNA molecule and start degrading them earlier. So here, the effects of the mutation in the polyadenylate polymerase enzyme is going to be the decreased stability or the decreased survival time of the actual mRNA molecule, because we're going to start degrading it faster due to the action of RNases and microRNAs. You'll also have to consider that the poly A tail is added on as part of your mRNA processing. And this will only occur inside of eukaryotes. So here, the mutation in polyadenylate polymerase is going to decrease the stability of your mRNA in eukaryotes, but because you don't have processing in prokaryotes, you're going to have no effect inside of your prokaryotes. So here, the best answer choice is going to be B, that you're going to decrease the stability of your mRNA molecule in eukaryotes because you're going to digest them by enzymes faster, whereas you'll have no effect in prokaryotes because they'd never add on this poly A tail anyways.